Hello. So very good evening to the student who have joined. Can you please let me know if my voice is audible to you? In the chat box, kindly let me know if you can hear my voice properly, and also please let me know if you can see the video. Hello. Am I audible? Please let me know if you can hear my voice, and also please let me know if you can see the video and if the screen is visible properly. Okay, thank you for confirming. Can you please let me know if you can see the presentation? The class PPT, is it visible to you properly or not? Okay, thank you so much for confirming. We will wait till it's 6 p.m. Then we will start today's session. Okay. Meanwhile, if you have any doubt or any query regarding our last session, if there is any thing you want to ask, you can let me know. We will start when it's 6 p.m. Okay, so till then, please wait. And meanwhile, if you have any problem regarding our week 10 live session, anything, any topic regarding our week 10 post content, you can please let me know because today it is our week 11 live session and we will start the session shortly. So if you have any doubt, you can let me know now. Okay. So let us wait till 6 p.m. Then we will start today's session. As I have already mentioned, if you have any doubt or any query, please let me know. So very good evening to the student who have joined. I hope my voice is audible and I also hope that you can see the presentation clearly. If not, please let me know. I hope everything is fine with the audio and also with the video. I have already mentioned that if you have any doubt or any query regarding our previous session, please let me know. Okay. We will start shortly once at 6 p.m. Then we will start today's session. Till then, kindly wait. So we will be starting within a minute. Okay. I hope everything is fine with the audio and video and presentation. If not, please let me know via the chat box. Okay. Okay, so it is 6 p.m. and I think I will be starting the session. So very good evening to the students who have joined. My name is Mousumi and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL SWAM course of Organic Chemistry in Biology and Drug Development. And today it is our week 11 live session. So before starting the session, let me clarify a few things. Okay, I hope the presentation is visible to you and my voice is also clearly audible. If not, please let me know through the chat box. So today it is our week 11 live session. So before starting the session, let me point out few things that there will be a problem solving session every week and these sessions are scheduled on every Friday from 6 to 8 p.m. The sessions are live on Google Meet platform. This is the link for joining the session which is scheduled on Friday from 6 to 8 p.m. So the important thing is if any network disruption happens from my side, if I have to leave in between the session, please you do not leave the session. Okay, I will try to rejoin as soon as possible, but you please stay on the call. And during the session, if you have any network issue from your side, you can just leave and rejoin at any time. The call is open, so you can rejoin at any time. Whenever you have the network issue, you can resolve it and you can rejoin back. The during the session, the students you can post your doubts and queries through the chat box. I am recording the session. This session recording and this presentation will be shared to you after the class. So since I am recording the session, I want it to be without any problem. That is why I have to mute your microphones and the cameras. But the chat box is always open, and you know that I monitor the chat box every now and then. So if you have any doubt or any query, you can let me know through the chat box. The chat box we will also use for our session throughout the session for communication for the especially for the question answering sessions. Okay, so how we will do that, I will let you know later. After the class, if you have any doubt or any query, you can let me know anytime during the session. And after the class also, if you have any doubts or queries, you can let me know via email. Okay. And my email ID I will share to you after the end of the session. Also, you can let me know via the discussion forum of the this SWAM course, which is available in the SWAM portal. Okay. And I have already mentioned that this presentation and class recording will be shared with you. Where to find this presentation and class recording? You have to go to NPTEL SWAM portal and you have to go to the course web page when there is this particular tab. Okay, the announcement tab is there about where the weekly course announcements and all the weekly stuffs are posted. Also here you are reminded for our every before every hour class, right? So here you will find there is a left panel where the lectures are, they are uploaded along with the assignments. And here you will find there is a problem solving session. If you go to this point, if you click on the problem solving session recording, you will be redirected to a page where you will find all the classes, the material, the class material, the presentation, the class recording, which is in form of the YouTube link. All it is available on that particular page. You have to just go and you have to you can the particular page will contain a Google Drive link like this, and this drive link will be, it will contain all the our weekly class PPTs whatever we have covered till now, and upcoming classes also it will be uploaded in this particular Google Drive. And you will find the YouTube link for the every class which you might have missed due to some reason. You can go through the lectures. Okay, after going through the lecture, if you have any doubts or any query regarding any class, you can let me know. Okay. So that is all what I have to say before starting the session. Now, what we do in our every live session, we actually a particular live session we have a particular week's course content, right? So in the last class, we have completed our week 10 course content, which has discussed regarding the antimicrobial agents, antibacterial agents. We have discussed about penicillin, different type of penicillins, their mechanism of actions, and so on. That we have discussed, and not only penicillins, we have discussed about various kind of our antibacterial drugs, antimicrobial drug, various families. We have discussed about the tetracyclines, we have discussed about the macrolides, we have discussed about the oxalidinon class of families, and all other mechanisms by which an antibacterial agent, a particular antibacterial agent can exert its antibacterial activity. That we have covered in our last class. Okay, so so today it is in our course content. So what are the course content of our week 11? Let us first see that. So our today's course contents are basically the polyketide biosynthesis. We have biosynthesis of the macrolide polyketide and introduction to virus, and we will discuss about the antiviral drugs. So antiviral drugs are basically and along with the polyketide biosynthesis is the topics which we are going to cover today. Okay, so like every class, what I will do is I will show you around 20 questions related to that particular week's course content regarding the week 11 course content, which was released in our last Friday. Okay, so that questions will be solely basically based on the week 11 course content. All the questions are MCQ type of question. All the questions have four different options. Okay, and after showing you a particular question, I will wait for some time, and you have to let me know A, B, C, or D, which of the following will be the correct answer for that particular question. Okay, whatever you say, I will check the answer first, and then I will discuss that answer in detail. Okay, so in the important thing is there is no marking here for answering the class. So even if your answer is wrong, it does not matter. Okay, you can answer with your free mind. Whatever you answer you feel is correct, you can let me know. Okay, there is no negative marking, so you don't have to worry about anything. But the assignments which are posted on the SWAM portal, the weekly assignments all have marks. So you have to be extra careful while answering those questions. Okay. So now let us start our today's session. I hope everything is fine with the audio and the video. If not, please let me know. Okay. So let me start with the first question of today's session. So the first question, the problem number one, it says that in the fatty acid biosynthetic pathway, conversion of 3-oxobutanone to 3-hydroxybutanone is catalyzed by dash. Mention the name of the enzyme. Your options are number A, beta ketoacyl ACP reductase, number B, beta hydroxyacetyl ACP dehydrase, number C, enoline ACP reductase, and number D, thioesterase. Which of the following enzyme is responsible for the conversion of 3-oxobutanoate to 3-hydroxybutanoate? You have to find out the particular enzyme which is responsible for the conversion of 3-oxobutanoate to 3-hydroxybutanoate. Okay. So from this 3-oxobutanoate to 3-hydroxybutanoate, this conversion is catalyzed by which enzyme? That you have to find out. Your options are number A, beta ketoacyl ACP reductase, number B, beta hydroxyacyl ACP dehydrase, number C, enoline ACP reductase, and number D, thioesterase. Which enzyme is responsible for the conversion of 3-oxobutanoate to 3-hydroxybutanoate? Let me know in the chat box. A, B, C, or D, which of the following you think is the correct answer? After checking your answer, I will show you the correct answer and we will discuss that particular question in detail. Okay. So let me wait for 2 to 3 minutes, then I will check the chat box. In the fatty acid biosynthetic pathway, conversion of 3-oxobutanoate to 3-hydroxybutanoate is catalyzed by which enzyme? There is no negative marking, so whatever you think is the correct answer, you can let me know without any hesitation. But you have to be extra careful while answering in our weekly assignments that are posted on the SWAM portal. Okay, because that particular questions have marks and negative marking also. Let me wait for another minute, then I will check the chat box. Okay, let me check the chat box now. Okay, I got two answers. One is saying it is number A and another saying it is number B. Okay, now let me check number A or number B, which of the following is the correct answer for our problem number of our first problem, A or number B. Okay, so the correct answer will be, it is number A, beta ketoacyl ACP reductase. Okay, now let me discuss it. Let me on just put the highlighter on. Yeah. So what are first, let us understand what are the functions of the fatty acids. So fatty acids, we all know, are very essential component and have a major physiological roles. Just like our protein and carbohydrate, fatty acids are also the major components which consist our body, or which consist the which actually makes all the fundamental elements of our body. So fatty acids are the building blocks for the phospholipid and glycolipids. Many proteins are modified by the covalent attachment of fatty acids, which target them to membrane locations. Fatty acids are the fuel molecules. They are stored as triglycerols, triacylglycerols. Fatty acid mobilized from the triacylglycerols are oxidized to meet the energy needs of a cell or organism. Fatty acid derivatives serve as the hormones and intracellular messengers, for example, steroids, sex hormones, and prostaglandins. Okay. Now this is that fatty acid, the conversion in the biosynthetic pathway. These are the important steps. Okay, so the first is the acyl ACP, which is a primer, and also the malonyl ACP, which is a chain extender. So these are the first components which together they form the 3-oxo butanoid. 3-oxo-butanoid is formed by these two components, and the enzyme which is responsible is the beta ketoacyl ACP synthase. Okay, so this is the enzyme which is responsible for the synthesis of the 3-oxo-butanoid, and then 3-oxo-butanoid is converted to the 3-hydroxybutanoid, and the enzyme which is responsible for this conversion is the beta ketoacyl ACP reductase. Okay, so beta ketoacyl ATP reductase is the enzyme which is responsible for the conversion of this 3-oxo-butanoid to 3-hydroxybutanoid. So as you can see, in case of 3-oxo-butanoid, you have, this is the first carbon, second carbon, third carbon. Here we have a keto group present in the 3-oxo-butanoid, whereas in the same position in the 3-hydroxybutanoid, it is reduced to the hydroxyl group. Okay, the CHO. And so this is the reduction kind of. So the reductase is the enzyme which is responsible for this reduction kind of reaction. And here it is the cofactor is the NADPH, and H plus is converted to NADP plus. So this is the source of the hydride, the NADPH. Then this 3-hydroxy-3-butanoid is converted to trans-2-butanoid, and the enzyme responsible is the beta-hydroxyacyl ACP dehydrase. Then this this double bond is trans here. Then this trans-2-butanoid is converted to only butanoid by aniline ACP reductase, and this after several steps it formed the type 1 fatty acid synthesis with the help of the thioesterase enzyme. Okay, so remember, in the biosynthetic pathway of fatty acid, 3-oxo-butanoid is converted to 3-hydroxybutanoid by beta ketoacyl ACP reductase. Okay. So is it clear, or you have any doubt? If you have any doubt, you can let me know. If in the middle of the class, if you have any doubt or any query, please let me know. Okay, during the session itself. Otherwise, I will go to our next problem if there is no doubt in problem number 1. Okay, let me go to the next problem. So we have covered it. So problem number 2, it says that the linking of the building blocks of polyketide biosynthetic pathway is similar to the reaction. So which is the reaction which is similar to this linking of this polyketide biosynthetic pathway. Okay, so the linking of building blocks in the polyketide biosynthetic pathway is similar to which reaction? Your options are number A, Claisen condensation, number B, aldol condensation, number C, Diels-Alder reaction, and number D, Michael addition. Number A, Claisen condensation, number B, aldol condensation, number C, Diels-Alder reaction, and number D, Michael addition reaction. Linking of the building blocks in a polyketide biosynthetic pathway is similar to which reaction? Your options: number A, Claisen condensation; number B, aldol condensation; number C, Diels-Alder reaction; and number D, Michael addition reaction. Let me know in the chat box A, B, C, or D which of the following you think is the correct answer for problem number 2. Again, I will wait for a minute or two, then I will check the chat box, okay? And then we will discuss the question in detail. The linking of the building blocks in a polyketide biosynthetic pathway is similar to which reaction? Claisen condensation, aldol condensation, Diels-Alder, or Michael addition? Which of the name reaction will be similar to this linking of the building blocks in polyketide biosynthetic pathway? Let me know A, B, C, or D which of the following you think is the correct answer for problem number 2. Okay, let me check the chat box first. Let me see. Okay, I got one answer saying it is number A. Okay, let me check if number A is the correct answer for our problem number 2. So the correct answer will be yes, it is number A. Claisen condensation is the reaction which is similar to the should say that linking of the building blocks in a polyketide biosynthetic its pathway is similar to the Claisen condensation reaction. So first, let us see what is the Claisen condensation reaction. I think if first we have to understand what is the mechanism of the Claisen condensation reaction. So the mechanism of the Claisen condensation reaction. Okay. Okay, let me just remove it. Okay. Yeah. So in the case of the Claisen condensation, we have this alpha proton has to be there. Okay, in the case of the particular beta, so it is a particular ester, right? So here we have what we have here. We have a first this alpha carbon, the proton it is deprotonated, and we have an anion by a base, which is a mild base, sodium methoxide. Then what happened? This particular anion it attacks on the similar the same molecule or same ester molecule but on the carbonyl carbon. Okay, once this negative charge attacks here, this double bond is goes on the oxygen forming the O minus, and then what will happen? This oxygen it will just come here, and this OET is a leaving group which will be leaving. So which we will have this which is known as a beta keto ester. Okay, so this is the alpha carbon, then is a beta carbon, and this is a ester, but it has a in the beta position it has a keto group. That is why it is known as a beta keto ester. Okay, so with the driving force of this reaction, the beta keto ester has a very acidic proton. Okay, which is basically removed by the base. Therefore, an acidic workup is required in case of this particular Claisen condensation. So this is the synthesis of a beta keto ester by the Claisen condensation. This is the mechanism. Now let us see what is the mechanism of the polyketide biosynthetic pathway. So it starts from the acetyl CoA. Okay, so this is the structure of acetyl CoA. I think in our previous one class we have discussed about this acetyl CoA and the complete synthesis, how it happens of everything we have discussed. So this acetyl CoA First is the carboxylation by carbon dioxide. So we have this similar to the Claisen condensation. We have this negative charge attacks on the keto group, and there is a I mean the negative charge because of this SCO group it is released and malonyl CoA is formed. Okay, so this step is similar to the Claisen condensation step, right? Then, so the first molecule of malonyl CoA is added to the premising malonyl fluoride, so which is a Claisen condensation reaction. And so then this kind of rearrangement happens, and the chain extension it keeps happen to form the poly beta keto ester. Okay. Then this poly beta keto ester, the folding happens and aldol condensation gives us the orselinic acid, which is a starting point or which is a very crucial intermediate in case of our polyketide biosynthetic pathway. Okay, so remember the name reaction which is similar or the linking of the building blocks of polyketide biosynthetic pathway is similar to our Claisen condensation reaction where the beta keto ester are formed. Okay. So, is there any doubt regarding this Claisen condensation, the mechanism, or anything? If you have any doubt, please let me know. Okay, I think it is a very simple mechanism. Only you have to practice it for several times. Okay, otherwise, this mechanism is very simple. Now let us go to the next problem. So problem number 3, it says that which one of the following statement is true regarding the primary metabolites. Your options are number A, they formed near the second stationary phase of the growth. Number B, they are not directly involved in the growth, development and reproduction. Number C, they are they are same in all plants. And number D, pigments are an example of primary metabolite. Which of the following statement is true regarding the primary metabolites? Again, I will repeat the options. Option number A, they form or they are formed near the stationary phase of the growth. Number B, they are not directly involved in the growth, development, and reproduction. Number C, they are same in all plants. And number D, pigments are an example of the primary metabolite. Which of the following statement is true regarding the primary metabolites? So wherever there is a true false type of question, I have mentioned it for a couple of in our previous classes also that the particular true false kind of question you have to pay extra attention because many students they tend to make mistake during this correct incorrect or true false kind of questions. Okay, so read all the options very carefully and then let me know in the chat box A, B, C or D which of the following you think is the correct answer for this problem number 3. Which of the following statements is true regarding the primary metabolites? Number A, they are formed near the stationary phase of the growth. Number B, they are not directly involved in the growth, development and reproduction. Number C, they are same in all plants. And number D, pigments are an example of the primary metabolite. Let me know A, B, C or D which of the following is the correct answer for problem number 3. I will wait for a minute, then I will check the chat box and we will discuss this particular question. Which one of the following statements is true regarding the primary metabolites? Let me check the chat box. Okay. Okay, I got one answer saying number A is the correct answer, means number A is true regarding problem in the problem number 3. Now let me check if it is correct or not. So the correct answer will be yes, it is number okay, what was the answer given in the chat box? Let me just check. Just forgot. Okay, the answer is given which is A, but the correct answer is actually number C. Okay. So primary metabolites are basically same in all plants. Okay, now let me show you, let me discuss you about the primary and secondary metabolites. So metabolism, it give rise to two type of metabolisms are there. Okay, one is primary and one is secondary. So primary metabolism is the general solution to biological problems necessary for growth, known physiological role active throughout the life cycle. It is ubiquitous and formation of a single product, and products are simple chemical structures. Okay, whereas secondary metabolites specific solution to biological problem, not required for growth, physiological role difficult to assess, present during the development stage, not ubiquitous, formation of families of products, and products are the complex of chemical nature. So actually what happens that the primary metabolites, they are basically same in all our compounds or our plants. Okay, but what happened, these are the secondary metabolites, they are very specific for the specific plants. Okay, they are not same in all the plants. So the primary plant primary metabolites are like antifungal activity, they show antiviral activity, antiseptic insect activity, and antibacterial activity. So primary metabolite, these are chemical compounds produced during the growth and development phase, not the stationary phase. Okay, so the statement number A has said that they are formed. Okay, it will be they are formed. Wait a second. So it has said that they are formed during the stationary phase. No, they are basically formed during the growth and development processes, right? So this is very important that these are formed during the growth and the development processes. They are also involved in the primary metabolic processes of respiration and photosynthesis. The primary metabolites are formed in the growth phase. They maintain a physiological functions of the body and are known as the central metabolites. They are intermediate products of the anabolic metabolism, which are used by the cells for the formation of the essential macromolecules. Amino acids, vitamins, organic acids, and some of the primary metabolites are produced industrially. Alcohol is the major primary metabolite that is produced on the large scale industrially. So primary and secondary metabolites. So the primary metabolites perform physiological functions in the body like intrinsic functions. Secondary metabolites are derivatives of the primary metabolites. They're directly involved in the growth, development, and reproduction, whereas the secondary metabolites, they play in the ecological functions. Primary metabolites are formed during the growth phase due to the energy metabolism, and for the secondary metabolism, these are formed in the stationary phase of the growth. Primary metabolites produced in the large quantities and extraction is easy. Secondary metabolites are produced in the small quantities and the extraction is difficult. Primary metabolites, they are same in all plants, whereas the secondary ones are unique in different plant species. Primary metabolites are a part of the basic molecular structure of an organism, whereas the secondary metabolites not a part of the basic molecular or structure of an organism. Ethanol, lactic acid, nucleotides, vitamins are examples of the primary metabolites, whereas pigments, antibiotic, and drugs are the examples of the secondary metabolites. Okay, so these are the differences between the primary and secondary metabolites. Now let us see the statements. So the first statement says that they are formed near the stationary phase of the growth. No, the secondary metabolites are formed near the secondary stationary phase of the growth, but this primary metabolites, they are formed during the growth and development process. Okay, thus they are not directly involved in growth, development, reproduction. It is also false. They are same in all plants. This is true, and pigments are not an example of the primary metabolites. So this statement is also false. Okay. So is there any doubt regarding our problem number 3? If you have any doubt or any query regarding primary or secondary metabolites, please let me know now. I will discuss it. Otherwise, I will move to the next problem. Okay, so I hope there is no issue. Then I will go to the next problem directly. So problem number 4, it says that what are the building blocks used in the biosynthesis of 6-deoxyerythritol B? Your options are number A, methylmalonyl CoA, methylmalonyl S-CoA and acetyl S-CoA. Number B, propanol S-CoA and acetyl S-CoA. Number C, malonyl S-CoA and methylmalonyl S-CoA. Number D, propanol S-CoA and methylmalonyl S-CoA. So what are the mainly the building blocks used in the biosynthesis of the 6-deoxyerythronolide B? You have to identify what are the correct building blocks for the 6-erythronolide 6-deoxyerythronolide B. Your options are number A methyl malonyl CoA, acetyl CoA, number B propionyl CoA, acetyl CoA, number C methyl malonyl CoA and methyl malonyl CoA, number D propionyl CoA and methyl malonyl CoA. What are the building blocks that are used in the biosynthesis of 6-deoxyribonucleic B? I will wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 4. What are the building blocks used in the biosynthesis of 6-deoxyribonucleic B? Let me check the chat box. Okay, I got an answer which is saying that number C is the correct answer. Let me check if number C is the correct answer or not for our problem number 4. So the correct answer will be, it is number D, propionyl CoA and methyl malonyl CoA. Okay. Okay. Mm, okay. So basically, this our 6-deoxyribonucleic B. So this is also known as the 6-DEB. Okay, so this is the short form. Okay, now so this particular this is a this is the starting points at this propionyl CoA and this methyl malonyl CoA. So this is these are the structures for this. This is the mixed structure of methyl malonyl. This is the structure for propionyl CoA. So the enzyme which is responsible is the deoxyribonucleic B synthase. Okay, so this let me just highlight it. So this is the enzyme which is responsible for the synthesis of the 6-deoxyribonucleic B, which is the deoxyribonucleic B synthase 1, 2, 3. Okay, so this is the structure. You do not need to memorize the structure, which is very cool because it is complex. So this is the start. This is actually this gives rise to the erythromycin A after several biosynthetic pathways. Okay, so this 6-deoxyribonucleic B or 6-DEB is a macrolide polyketide serving as the aglycone precursor to the important macrolide antibiotic erythromycin. So what is the structure? It is a 14 membered, so it is a 14 member macrolactone with the molecular formula of C21H38 and O6 and a molecular weight of the 386.5 gram per mole. Its complex structure includes the multiple stereocenters and is characterized by the macrolactone ring, which is central to its biological activity. Biosynthesis 6-DEB is synthesized in nature by 6-deoxyribonucleic B synthase or 6-DEBS, a large modular polyketide synthase enzyme complex found in the bacterium Saccharopolyspora erythritria. The biosynthesis involves a series of enzymatic steps, including the sequential chain elongation using the propionyl CoA as the starter unit and methyl malonyl as the extender units, following the reduction and the macronolactonization to form a 14-membered ring. What is the significance? The 6-deoxyribonucleic B is a crucial intermediate in the industrial production of the erythromycin and its derivatives, which are widely used antibiotics. The study of the DBS and 6-DEB biosynthesis has provided valuable insights into the mechanism of the modular PKS enzymes and has opened the avenues for the engineering of these systems to produce novel polyketides with a therapeutic potential. Okay. So this is the about the 6-deoxyerythronolide B. The importance, their significance that you have to remember. The structure is little complex, so you do not need to memorize the structure. So I hope it is all right. If there is any doubt, I am saying repeatedly that if you have any doubt during any question, please let me know in the chat box. I will discuss that particular question at that time itself. Okay. Let us go to the next problem then. If there is no doubt, the problem number 5, it says how many modules are involved in the formation of the macrolide core of erythromycin, the macrolide core in the erythromycin biosynthesis. How many modules are involved in the formation of the macrocyclic core in the erythromycin biosynthesis? Their options are number A 7, number B 8, number C 6 and number D 3. How many modules are involved in the formation of macrocyclic core in the erythromycin biosynthesis? 7, 8, 6 or 3? How many modules are involved in the formation of the macrocyclic core in the erythromycin biosynthesis? 7, 8, 6 or 3? I will wait for a minute, then I will check the chat box. How many modules are involved in the formation of macrocyclic core in the erythromycin biosynthesis? Let me check the chat box. Okay, I got an answer which is saying that number A is the correct answer. Let me check if it is so. So the number of modules that are involved in the formation of the macrocyclic core in the erythromycin biosynthesis will be, it is, the correct answer is number C, which is number 6. So 6 modules are there. Okay. So erythromycin biosynthesis, specifically the formation of its polyketide backbone, the 6-DEB, which I have discussed in the last slide itself, that erythromycin polyketide backbone, the 6-deoxyerythronolide B, involves 6 modules within the 6-deoxyerythronolide B synthesis enzyme complex. Each of these modules are responsible for one round of polyketide chain extension and modification. The subsequent tailoring modifications to convert 6-DEB to mature erythromycin A involves other enzymes like hydroxylases, glycosyl transferase and methyl transferase, but these are not considered as a part of the PK system itself. Okay, so this is the synthesis of the erythromycin biosynthesis. So I have mentioned in the last that first the succinate is converted to the succinyl CoA, okay, and then it is converted to the 2R methyl mannoline CoA, and we have the propionyl CoA also here we have, and then we have this 2S methyl mannoline CoA which is formed. Okay, and then we have the 4 system, 3 complexes of this DEBS, which is the DEB synthase. Okay, then this enzyme, the D6-deoxyerythronolide synthase we have. This is the DEBS1, DEBS2 and DEBS3. Each have two modules each. So we have the 6 modules in total, and this is a little bit complex. You do not have to memorize this. If it is possible, you can memorize, but it is not very much necessary. Then after that, the 6-deoxyerythronolide B is formed, which forms this erythromycin C and erythromycin D. Okay, so the backbone is this formation of the backbone. It actually involves this six more number of modules. Okay, so this was the correct answer. Now I will go to the next problem, problem number 6, which says that which of the following statements is true regarding the virus? Which of the following statements is true regarding the virus? Again, it is a true false type of question, so read all the options very carefully and identify the statement which is true regarding the virus. Your options are: number A, viruses are not living organisms; number B, viruses grow and reproduce outside the host cell; number C, tuberculosis is a viral disease; number D, viruses are giant compared to the bacteria. Which of the following statements is true regarding the virus? Your options are: number A, viruses are not living organisms; number B, viruses grow and reproduce outside the host cell; number C, tuberculosis is a viral disease; number D, viruses are giant compared to the bacteria. Which will be the which statement will be true regarding the virus? A, B, C, or D? Again, I will wait for a minute, then I will check the chat box. Meanwhile, let me know A, B, C, or D, which of the following is the correct answer for problem number 6. Which of the following statements is true regarding the virus? Your options are: number A, viruses are not living organisms; number B, viruses grow and reproduce outside the host cell; number C, tuberculosis is a viral disease; and number D, viruses are giant compared to bacteria. You have to identify the statement which is true regarding the virus. Let me check the chat box. Okay, I got two answers. One is saying number D is correct and another is saying number A is correct one. Let me check which of the following is the correct answer for problem number 6. So the correct answer will be number A, viruses are not living organisms. This statement is true regarding the virus. So what are the key differences between the virus and bacteria? Okay, so viruses are not living organisms, but bacteria are living organisms. Viruses only grow and reproduce inside the host cell. They infect when found outside these living cells. Viruses are dormant. Their life therefore requires hijacking the biochemical activities of a living cell. Bacteria is a living organism that consists of a single cell that can generate energy, makes its own food, move and reproduce, typically by binary fission. This allows bacteria to live many places like soil, water, plants and human body and serve many purposes. Viruses are submicroscopic. Bacteria are giant compared to viruses. A viral infection is systematic. Viruses infect a host cell, then multiply by thousands and leaving the host cell, infecting the other cells of the body. Whereas bacterial infection is usually confined to a part of a body described as a localized infection. Viruses may be the infection may be caused by the bacteria or by the toxin like endotoxins produce. Systematic diseases caused by viral infection include influenza, measles, polio, AIDS, and COVID-19. Bacterial diseases include the pneumonia, tuberculosis, tetanus, and food poisoning. So what are the general characteristics of the virus? This is very important. Okay, what are the general characteristics of the virus? So viruses are generally obligate, intracellular, unable to self-replicate. So this is very important. Okay, the obligate, intracellular, and they are unable to self-replicate. Once inside the living cell, viruses induce a host cell to synthesize virus particles. The genome is either DNA or RNA, single or double-stranded. Viruses do not have the system to produce ATP. Okay, that is why these are not the living organism. Viruses range in size from 25 to 270 nanometer. viral tropism. So the classification of virus based on the nucleic acid type, size, and shape of the virion, presence and absence of an envelope, viral structure, virion, and entire viral particle. Capsid is the protein code that encloses the genetic material. A capsomer is the protein subunit that makes up the capsid. Nucleoplast capsid is composed of the capsid and genetic material. The envelope is the outer coating, which is composed of the phospholipid bilayer, which is composed of the viral encoded glycoprotein and sometimes viral encoded matrix protein. The envelope is derived from the host cell's membrane. Some viruses use the plasma membrane, whereas others use endoplasmic reticulum, Golgi body, nuclear membrane, naked nucleocapsids, and viruses with no envelopes. Okay. So these are the general characteristics of the virus. So the statement which says viruses are not living organism, this is true. Okay. Now let me go to the next problem. So problem number 7, it says that identify the correct order of a viral infection in the host cell. Your options are number A, attachment, followed by uncoating, followed by penetration, followed by disease release, followed by assembly, followed by biosynthesis. Your option number B is first attachment, then is penetration, then uncoating, then biosynthesis, then release, then assembly. Number C is the attachment, followed by biosynthesis, followed by penetration, then uncoating, then assembly, and then release. And number D, attachment, followed by penetration, followed by uncoating, then biosynthesis, then assembly, and then release. So which is the correct order for the viral infection in the host cell? So these are the different steps, but you have to identify what is the correct order of the viral infection in the host cell. I will wait for a minute or two, then I will check the chat box. Meanwhile, let me know which of the following will be the correct order for the viral infection in the host cell. Identify the correct order of the viral infection in the host cell. So, what will be the correct order for the viral infection in the host cell? Let me check the chat box. Okay, I got one answer saying number C is the correct answer. Let me check. Let me check if number C is the correct answer for problem number 7. So the correct answer will be, the correct order for the viral infection in the host cell will be, it is D. First is the attachment, followed by the penetration, then uncoating, then biosynthesis, then assembly, and then release. Okay. Now let us discuss it in detail. So what are the steps for the viral infection? I have already mentioned that the steps and the order is the attachment, penetration, uncoating, replication, assembly, and release. Once inside the host cell, the viral DNA or RNA is released and can begin to interact with the internal cell machinery. Viral DNA can direct transcription of the viral mRNA, which is then translated into the viral proteins. Viral RNA can be used as a template to make more viral mRNA and proteins. RNA retroviruses use the reverse transcription to make the viral DNA, which is then transcribed and translated. Regardless of the mechanism, the objective is the same, more virions. New virions are released when damage to the host cell causes lysis, or when the host cell undergoes the apoptosis or programmed cell death. Some viruses are released through the budding, which releases one virion at a time that spurs the host cell. So this is the representation, the life cycle of our DNA virus, for example, as the herpes simplex. Okay. So what is the steps? The first step is the absorption or the attachment. So first this virion, it is attached with the host cell membrane. Okay. Then form this the penetration. So the penetration happens by fusion when this virion is taken up inside the cell. Then we have what is which happens is after that we have the step number C, which is the first is the uncoating happens. Okay, the uncoating means now the nucleoplastic or the viral in this particular viral DNA is goes inside the nucleus and which forms the viral mRNA. This viral mRNA forms the viral which is the uncoated biosynthesis of the replication and transcription. Okay, and then viral nucleoplastic or which is happens, which is the synthesis of the assembly of the nucleoplastics, and then new viral formations, new viral virions are formed, and then it's the assembly. After the assembly, the step is the budding and the viral, which is and the release. When the new virion is released outside the host cell. Okay, so these are the different steps and these different life cycles and the different steps for the viral infection. Okay. So is it all right or is it clear or I have to repeat it? Please let me know if it is fine or should I repeat it or anything. This is very easy. Just you have to see the figure properly and everything is mentioned via the figure which is very clear in detail. So if you have any doubt, let me know. Otherwise, I will move forward. So I hope it is clear how this particular viral infection happen via these steps of the attachment penetration, uncoating, attachment penetration, uncoating, replication, assembly, and release. Okay. Okay. So if there is no doubt, let me go to our next problem. So problem number 8 says which of the following statements is not true? Again, we have a true false kind of question, so you have to be extra careful. So number A, DNA virus has lower mutation rate. Number B, retroviruses are RNA viruses. Number C, RNA virus mutation occurs inside the nucleus. And number D, double-stranded DNA viruses are most common. Which of the following statement is not true? Which of the following statements is not true? Here you have to check that here it is mentioned that which of the following statement is not true. Okay, so you have to identify the incorrect statement among the following. Your options are number A, DNA virus has a lower mutation rate. Number B, reoviruses are RNA viruses. Number C, RNA virus mutation occurs inside the nucleus. Number D, double-stranded DNA viruses are most common. Let me know A, B, C or D which of the following is the correct answer for problem number 8. I will wait for a minute. Then I will check the chat box. Which of the following statement is not true? DNA virus has a lower mutation rate. Number A, number B, reovirus or RNA virus. Number C, RNA virus mutation occurs inside the nucleus. Number D, double-stranded DNA viruses are most common. A, B, C or D, which of the following statement is not true for problem number 8? Let me know in the chat box. I will wait for a minute, then I will check it. So, whatever you think is the correct answer, let me know. Which of the following statement is not true? Let me check the chat box now. Okay, got an answer. I got two answers, and both are saying that number C is the correct answer. Means number C is the statement which is not true. Let me now check if it is correct or not. So the correct answer, the statement which is not true will be yes, it is number C. Very good. RNA virus mutation occurs inside the nucleus. This statement is not true. So, what are the differences between the RNA and DNA virus? Let us check from here. So, the DNA viruses are the viruses that possesses the DNA as their genetic material. Okay. And RNA viruses are the viruses that possesses the RNA as their genetic material. The common type, most common type is the double-stranded DNA viruses, and the RNA viruses, most common type is the single-stranded RNA viruses. Genome size, DNA viruses, the genome size is larger, and the RNA viruses, the genome size is smaller. Replication, it occurs inside the nucleus, and the replication of the RNA virus, it occurs in the cytoplasm. Okay, so inside the nucleus for the mutate replication for the DNA virus occurs inside the nucleus, but in case of RNA viruses, this occurs inside in the cytoplasm. Mutation rate, lower mutation rate is for the particular DNA viruses. RNA viruses have a higher mutation rate. Accuracy of replication, slow and accurate replication happens in case of DNA viruses, whereas slow and error-prone replication happens in RNA viruses. Types, double-stranded DNA viruses and single-stranded DNA viruses are the most common one. Double-stranded RNA viruses, positive sense single-stranded RNA viruses, and negative sense single-stranded RNA viruses is the most common one. Adenovirus, herpes virus, pox virus, parvo viruses, and hepatoviruses are the example of DNA viruses, whereas reovirus, piconar virus, toga viruses, orthomyxoxid viruses, and retroviruses are the example of RNA viruses. Okay. So DNA virus, these are the examples, which are the smallpox, herpes, okay, chickenpox, adenovirus like sore throat and conjunctivitis, hepatinoviruses like serum hepatitis, and papilloma viruses, which is the warts. Whereas the RNA viruses like myxoviruses like influenza, measles, mumps, like viruses, German measles, rabies, cold, meningitis, poliomyelitis, AIDS, T-cell leukemia, meningitis, Lassa fever, and arthropod-borne encephalitis, yellow fever. These are happened. This happened due to the RNA viruses. So, is it clear what is the differences between the DNA and RNA viruses? Is there any doubt? If there is any doubt regarding the differences between the DNA and RNA viruses, please let me know. Otherwise, I will move to our problem number 10 if there is no doubt. Okay, problem number 9. Okay, so I think it is clear. Then I will let me go to the next problem. So problem number 9, it says that which of the following enzyme is responsible for incorporating viral DNA with host DNA? Your options are number A, reverse transcriptase, number B, protease, number C, integrase, and number D, DNA polymerase. Which of the following enzyme is responsible for incorporating viral DNA with the host DNA? Your options are number A, reverse transcriptase, number B, protease, number C, integrase, and number D, DNA polymerase. Let me know A, B, C, or D, which of the following will be the correct answer for our problem number 9. Which of the following enzyme is responsible for incorporating the viral DNA with the host DNA? Reverse transcriptase, protease, integrase, or DNA polymerase? We'll wait for a minute, then check the chat box. Let me know A, B, C, or D, which of the following you think is the correct answer for problem number 9. Which of the following enzyme is responsible for incorporating viral DNA with host DNA? Let me check the chat box. Okay, I got one answer saying number A is the correct answer. Let me check if number A is correct answer or not. So the correct answer will be, it is C, integrase. Integrase is the enzyme which is responsible for incorporating the viral DNA with the host DNA. Okay, so this is this particular structure for a HIV enzyme. Now let's just, so this is the structure of a HIV particle. Okay, this is the structure. These are the first is there is a enzyme which is called the integrase. We have enzyme also which is known as the reverse transcriptase. It is the viral envelope. Then here we have a capsid which actually covers this viral genomic material, and we have several other things here. So this is the whole. So this thing is almost similar to our which we have seen earlier also. So the steps are similar. So integrase is the enzyme which actually facilitates the facilitates the integration of the viral genetic material into the host cell DNA. The process is the critical step in the replication cycle of the retroviruses such as the HIV. So the steps are, first is the absorption. Just I have mentioned earlier also. So first the virions are absorbed, and it is by fusion inside the cell. So inside the cell it actually have this viral RNA which is converted to viral DNA or DNA hybrid by the viral RNA DNA hybrid by the reverse transcriptase. Okay, so the reverse transcriptase is the enzyme which is responsible for the conversion of the RNA to the RNA DNA RNA hybrid, which forms a single strand DNA. This single strand DNA by the reverse transcriptase also form the DNA, and then integrase is the enzyme. What integrase does, it actually, so as you can see, the host DNA is mentioned in this black, and this is the cyan one is the viral DNA. So integrase is the enzyme which incorporates this viral DNA and the host DNA together, which forms then the RNA. Okay, this RNA now it consists the viral particle, the viral genome particle genome also with the host. Now, so then it forms a polyprotein and the protease, and the viral proteins are synthesized, which will generate new viral particle. Okay, so integrase is the enzyme which facilitates the integration of the viral genetic material into the host cell DNA. The process is a critical step in the replication cycle of the retroviruses such as HIV. How the integrase work? The integrase in the integration process catalyzed by integrase within the large protein DNA complex called the intasome occurs in two main steps. First is the 3' processing. Okay, after the viral RNA genome has been converted into the double-stranded material by double-stranded viral DNA by by enzyme reverse transcriptase, the integrase clips 2 or 3 nucleotides from the 3' end of the each strand of the viral DNA. This exposes to the crucial pair of the 3' hydroxyl group. Second, and then since the strand transfer happen, the processed 3' ends of the viral DNA are covalently ligated to the host chromosomal DNA. The integrase makes a staggered cut in the host DNA, allowing the viral DNA ends to be inserted. The resulting gaps are then repaired by the host cell enzymes, completing the integration. Okay, so these are the how integrase work. These are the two main steps by which this integrase enzyme works. And integrase enzyme, let me tell you, it is a very good drug target for the anti-HIV drug discovery. Okay, it is a very good target, the integrase enzyme. So is it clear how integrase works and what it does basically? So I hope it is clear. If you have any doubt, you can let me know in the chat box. As I have mentioned that in case of the HIV, there are several enzymes which are important. Integrase is one of them, and it is a very good drug target for our anti-HIV drug discovery. Okay. So if it is clear, if there is no doubt, let me go to the next problem of today's session. So problem number 10, it says identify the incorrect statement. Okay. So you have to here identify the statement which is incorrect. So again, it is a correct incorrect kind of question which for which you have to be extra careful. So just let me just show you. So number A, NRTIs incorporate into the viral DNA and blocks the transcription. Number B, NNRTIs are non-competitive inhibitors. Number C, NNRTs NNRTIs, okay, it will be NNRTI, okay. NNRTIs bind with the reverse transcriptase. Number D, NRTIs are non-competitive inhibitors. Again, I am repeating, identify the statement which is incorrect. Options are number A, NRTI incorporate into the viral DNA and blocks the transcription. Number B, NNRTIs are the non-competitive inhibitors. Number C, NNRTIs bind the reverse transcriptase. And number D, NRTIs are non-competitive inhibitors, okay. So, which of the following statement is incorrect? Let me tell me in the chat box. A, B, C, or D. Which of the following statement is incorrect one? Your options are number A, NRTI incorporate into the viral DNA and blocks the transcription. Number B, NNRTIs are non-competitive inhibitors. Number C, NNRTIs bind with the reverse transcriptase. And number D, NRTIs are non-competitive inhibitors. Again, I will wait for some time. I will wait for a minute or two, then I will check the chat box. Let me know A, B, C, or D, which of the following statement is incorrect. Identify the statement which is incorrect. Number A, NRTIs incorporate into the viral DNA and blocks transcription. Number B, NNRTIs are non-competitive inhibitor. Number C, NNRTIs bind with the reverse transcriptase. And number D, NRTIs are non-competitive inhibitor. Let me check the chat box. Let me see if there is any answer for problem number 10. Okay, I got two answers. One is saying number B is the correct answer and one is saying number D. Let me check number B or number D, which will be the correct answer for our problem number 10, right? Okay. So the correct answer means the statement which is incorrect will be, it is number D. NRTIs are non-competitive inhibitor. This is the incorrect statement. Okay. So what are the difference between the NRTIs and NNRTIs? So NRTIs, which are the class of the drugs that inhibit that HIV-1 reverse transcription enzyme by competing with the natural nucleosides. Okay, such as the DTTP, DCTP, GGTP, and DATP. And NNRTIs are the small molecule drugs that bind directly to the active site of the HIV-1 reverse transcriptase. NRTIs incorporate into the viral DNA and blocks the transcription, whereas NNRTIs binds to the reverse transcriptase, changes the special configuration of the enzyme. NRTIs are the competitive inhibitor, whereas NNRTIs are the non-competitive inhibitor. Example: lamivudine, emtricitabine, disoproxil fumarate, and tenofovir alafenamide. These are the examples of the NRTIs. And delaverdine, entravirine, nevirapine, nevirapine, doloravibrapine, and rilpivirine are the example of the NRTIs. Okay. So NRTI, how the NRTI works? So prodrug activation, the most NRTIs are administered via the prodrug, meaning they are inactive until they are taken up by the host cells and undergo phosphorylation by the cellular kinases to become the active phosphide forms. These are the competitive inhibitor. The activated NRTI structurally resemble the natural nucleoside triphosphates of the HIV reverse transcriptase enzyme uses to synthesize viral DNA from its RNA template. Chain termination: when the reverse transcription enzyme incorporates into an NRTI into the growing viral DNA chain, the process of the DNA synthesis is terminated. This is because NRTIs lack the crucial 3' hydroxyl group required for forming the phosphodiester bond with the next incoming nucleotide, thus preventing further elongation of the viral DNA, preventing replication by inhibiting the reverse transcription process. NRTIs effectively prevent the HIV from making copies of its genetic material by integrating it into the host cell's DNA, thereby halting the viral replication and slowing down the progression of the HIV infection. Examples of the NRTI include the abacavir, emtricitabine, lamivudine, tenofovir, and zidovudine. So these are the examples of the NRTIs. Okay. So this NRTI and NNRTIs are a very crucial anti-HIV drugs. Okay, very crucial class of the anti-HIV drugs. So is it clear, or do I need to repeat it? What is NRTI? What is NNRTI? So this NRTI and NNRTI, as I have mentioned, they are very, crucial anti-HIV drugs. Okay, because these are the reverse transcription enzyme inhibitors and the reverse transcriptase inhibitors. And reverse transcriptase are a very crucial enzyme in the life cycle of HIV virus, right? And as I have mentioned in the earlier slide also, that the integrase is a very good drug target. So as the protease, which is also a very good drug target, and reverse transcriptase. So this nucleoside reverse transcriptase inhibitor and non-nucleoside reverse transcriptase inhibitors are very crucial classes of the anti-HIV drugs. Okay, and these are the examples and these are the comparisons. So if there is no doubt, I will go to our next problem. So problem number 11 says, which of the following is not an example of NRTIs? Which of the following is not an example of nucleoside reverse transcriptase inhibitor? Your options are: number is zidovudine, number B abacavir, number C nevirapine, and number D stavudine. If you have paid attention in our last slide, I know you can answer this question very easily and correctly, because I think I have mentioned in our last slide only. Which of the following is not an example of NRTIs? Zidovudine, abacavir, nevirapine, or stavudine? Let me know A, B, C, or D. Which of the following will be the correct answer? I will wait for a minute only, because already I have mentioned in our last, I think in our last slide, I have already mentioned a couple of names of the nucleoside reverse transcript inhibitor and non-nucleoside reverse transcript inhibitors. Let me see if you have paid attention to the last slide. Only I will wait for a minute and then I will check the chat box. Okay. Which of the following is not an example of NRTIs? Let me check the, okay, let me check the chat box first. Okay, I think the answer is for this question. No, the answer I think is for the last question. Okay. Okay, let me check. There is no answer, but let me check what is the correct answer. So the correct answer will be, it is number C. Nevirapine is not an example of the NRTI. Okay. So nucleoside reverse transcriptase inhibitors, or NRTIs. Just let me use the highlight here. So nucleoside reverse transcriptase inhibitor or NRTIs. These NRTIs are the currently used as a part of the combination therapy for combating HIV. Generally, NRTIs have good oral bioavailability, are bound minimally to plasma proteins and are excreted through the kidneys. They also are act against both HIV 1 and 2. However, they are often associated with toxic side effects. Zirobudin was the first anti-HIV drug to reach market. This is important. Okay, that Zirobudin has is the first HIV drug to reach the market, but it has a severe side effect like anemia. Okay, Titonicin was the second anti-HIV drug approved for the use reached in the US market in 1988. However, there is a risk of the toxicity to the pancreas. Abacavir was approved in 1998 and has been used successfully in children in combination with the protease inhibitors nalfiravir and saquanavir. However, life-threatening hypersensitivity reactions have been reported in the same in some patients. Tenofovir disoproxil was approved for HIV-1 treatment in 2001. It remains in infected cell longer than many other antiretroviral drugs, allowing for once a daily dosing, and it can have allowed for once, but it can have toxic side effects on the kidney. It can be used alongside the emtricitabine, which was relatively free of the toxic side effect. Other NRTIs used against HIV include the lamivudine, stavudine. Lamivudine is less toxic than zirobudine and has also been approved for the treatment of hepatitis B. And zalcitabine is a NRTI which acts against the hepatitis B, but long-term toxicity means it is unacceptable for the treatment of the chronic viral diseases which are not life-threatening. Adefovir diprivoxil was approved by the US Food and Drug Administration or FDA in 2002 and the treatment of the chronic hepatitis B. It is also active in the viruses such as the CMV and herpes. And then we have non-nucleoside reverse transcriptase inhibitor or NNRTI. Compared with the NRTIs, the NNRTIs show the higher selectivity for HIV-1 reverse transcriptase over the host DNA polymerase. As a result, NNRTIs are less toxic and have fewer side effects. Unfortunately, rapid resistance emerges if the NNRTI is used on its own, but it does not occur if the NNRTI is combined with the NRTI from the start of the treatment. NNRTIs are restricted to the HIV-1 activity and are generally metabolized by the liver. They can interact with the other drugs and bind more strongly to the plasma protein. Nevirapine, efavirenz, delavirdine, etranavir, rilpanavir, and NNRTI are currently approved by FDA for the treatment of the HIV. Okay, so this is all about the NRTI and NRTI. Okay, so let me go to the next problem. If you have any doubt or query, please let me know through the chat box. Okay. So problem number 12 says the HIV protease enzyme is symmetrical whereas the mammalian proteases are not. What significance might this have? Your options are number A, inhibitors of the HIV protease enzyme must also be symmetrical. Number B, it may be possible to design inhibitors that prove selective for HIV protease over mammalian protease. Only symmetrical substrates are cleaved by the HIV protease. Number D, there is no significance. HIV proteases enzyme is symmetrical whereas mammalian proteases are not. What significance might this have? Your options are number A, inhibitors of HIV protease enzyme must also be symmetrical. Number B, it may be possible to design inhibitors that prove selective for the HIV protease over mammalian protease. Number C, only symmetrical substrates are cleaved by HIV protease. And number D, there is no significance. Let me know A, B, C or D, which of the following will be the correct answer for problem number 12. I will wait for a minute or two, then I will check the chat box. Okay, let me know. The HIV protease enzyme is symmetrical, whereas the mammalian proteases are not. What significance might this have? Think and then let me know A, B, C, or D, which of the following will be the correct answer. Okay, let me just check the chat box because I have to discuss it. Okay. So is there any answer for problem number 12? Okay, I got one answer saying number B is the correct answer. Let me check if number B is the correct answer or not. So the correct answer will be yes, it is number B. It may be possible to design inhibitors that prove selective for the HIV protease over the mammalian protease. So HIV protease enzyme is symmetrical dimer made up of the two identical protein units, each consists of 99 amino acids. The active site is at the interface between the protein units are also symmetrical with the two-fold rotational or situ symmetry. The enzyme has a broad substrate specificity and can cleave a variety of peptides bond in the viral polypeptides, but crucially it can cleave bonds between a proline residue and an aromatic residue like phenylalanine or tyrosine. Moreover, the symmetrical nature of the viral enzyme and its active site is not present in the mammalian proteases, again suggesting that there is a possible of drug selectivity. So whenever we are designing a particular drug for a very particular disease, let's say we are designing an antiviral drug for a virus, for combating a virus, and we are using a particular enzyme like for the targeting or we are trying to inhibit a particular enzyme of that particular of the virus. Okay, but if the similar enzyme is present in the host cell also, so it is crucial to have selectivity to achieve selectivity so that our drug, whatever we have designed, whatever the drug we have made, it so that it kills or it inhibits the protease enzyme of the virus, not the host cell. Okay, so the symmetrical structure of the HIV protease enzyme, which is the not the structure is not similar to the host mammalian proteases, is a very good thing because it helps helps us to design an inhibitor which is specified for the symmetrical nature of the viral enzyme and for active site, not the mammalian enzyme. Okay. So this is the significance which this particular phenomena might have. Okay. So drug selectivity is another very crucial issue. Okay, because now, whenever you are designing a drug, you must achieve the selectivity. Specifically, in cases of that, the drug is targeting the particular hosts. I mean, particular enzymes of the virus or bacteria or any other parasite or so. Okay. Okay, so it is the problem number 12. So yes, number B was the correct answer. Let me go to the next problem. Problem number 13 says, on which receptor of T cell surface the HIV binds? On which receptor of T cell surface the HIV binds? Your options are number A CD4, number B CD6, number C CD7 and number D CD2. On which receptor of T cell surface the HIV binds? If you have paid attention to our last slides, a couple of slides earlier, when I was discussing the structure of the virus, I have mentioned it, I think. So, on which receptor of the T cell surface the HIV binds? CD4, CD6, CD7, or CD2? I will again wait for a minute or two, then I will check the chat box. Okay, let me know whatever you think is the correct answer on which receptor of T cell surface the HIV binds. On which receptor of T cell surface the HIV binds. On which receptor of T cell surface the HIV binds? CD4, CD6, CD7 or CD2? Let me check the chat box. Let me see if there is any answer for our problem number 13. Okay, I got one answer saying number C is the correct answer. Let me check if number C is the correct answer or not. The correct answer will be, it is number A, CD4. I think I have mentioned it earlier also. Okay, so the role in the HIV infection, the CD4, the HIV hijacks the CD4 receptor to infect and ultimately destroy T helper cell, leading to the immune deficiency that characterizes the AIDS. Okay, so binding the HIV envelope protein, the GP120 binds directly with a high affinity to the CD4 receptor on the T helper cell surface. Okay, then co-receptor engagement, this binding causes the conformational change in the viral protein, allowing it to bind to the second co-receptor of the host cell, either CCR5 or CXCR4. Fusion and entry, the dual receptor binding that facilitates the fusion of the viral envelope with the host cell membrane, allowing the virus to enter the cell. Immune depletion, as HIV replicates and kills CD4 plus T cells, the body's immune system is progressively weakened and making it susceptible to opportunist infections. Monitoring the CD4 count is a standard part of the HIV treatment to assess the health of the patient's immune system. This is very important, that monitoring the CD4 count is a standard part of the HIV treatment to assess the health of the patient's immune system. Okay, so this is the HIV virus. As you can see, this is the HIV virus which has several components like integrase enzyme, which has RNA reverse transcriptase, capsid protease enzyme, and we have these receptors like GP41 and GP120. So the main is the GP120, which binds with the CD4, and the co-receptors like CCR5 and CXCR4. Okay, then after binding, what happens? So the first step is the binding on the surface of the T cell. HIV binds with the CD4 receptor and one of the co-receptors CXCR5 or CCR5. Okay, then the second step is the fusion. The virus fuses with the host cell membrane, releasing the genetic material or RNA into the cell. As you can see, RNA is released in the cell, then RNA with the help of the reverse transcriptase for the with the single stranded HIV RNA is converted to the double stranded HIV DNA by the reverse transcriptase enzyme. Okay, then the fourth step is the integration step. After HIV DNA enters the cell's nucleus, the enzyme integrase cuts the cell's DNA and insert the HIV DNA into it. Then the fifth step is the transcription and translocation. The RNA polymerase makes the RNA copies of the DNA. HIV RNA is either inserted into the new virus particle or processed by the translated into the HIV protein. Then is the assembly. Long protein chains cut into the individual protein to form HIV protease. The new virus assemblies assembled with these proteins and HIV RNA, and then release new virus particle is released into the host cell, taking it with a part of the cell's membrane and capable of infecting the other cells. Okay. So this is the life cycle of a HIV virus, which is similar to the our general life cycle, but there are several things which are very important. So which you have to identify and understand. So CD4 is a receptor on the T cell surface where this particular our HIV binds. Okay, so this was our problem number 13. Now let me go to the next problem. Problem number 14 says, which of the following statements is true regarding the HIV protease enzyme? Your options are: number A, it belongs to the family of the cysteine protease. Number B, it catalyzes the formation of the peptide bonds. Number C, active site structure contains the cysteine moiety. And number D, active site structure contains the aspartic acid. Which of the following statements is true regarding the HIV protease? Your options are: number A, it belongs to the enzyme family of cysteine protease. Number B, it catalyzes the formation of the peptide bonds. Number C, active site structure contains the cysteine moiety. Number D, active site structure contains the aspartic acid. Which of the following statements is true regarding the HIV protease enzymes? Again, it is a true false type of question. So read the options very carefully and let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 14. Which of the following statements is true regarding the HIV protease enzyme? Which of the following statements is true regarding the HIV protease enzyme? Number A, it belongs to the enzyme family of cysteine protease. Number B, it catalyzes the formation of the peptide bonds. Number C, active site structure contains the cysteine moiety. Number D, active site structure contains the aspartic acid. Let me now check the chat box. Let me see if there is any answer given in the chat box for our problem number 14. Okay, I think I do not have any answer for our problem number 14. Okay, it's fine. Let me check what is the correct answer and let us discuss about it. So the correct answer will be, it is number D. The active site structure contains aspartic acid. This statement is true regarding the HIV protease enzyme. So HIV protease enzyme is an example of an enzyme family called the aspartyl protease, the enzymes which catalyze the cleavage of the peptide bonds and which contain an aspartic acid in the active site that is crucial to the catalytic metabolism mechanism. The enzyme is relatively small, can be obtained by the synthesis, and alternatively it can be cloned and expressed in fast growing cells purified in large quantities. The enzyme is crystallized with or without an inhibitor bound to the active site, making it an ideal target for the structure-based drug design where the HIV protease enzyme is symmetrically dimer made up of the two identical protein units, each consisting of 99 amino acids. The active site is the interface between the protein units and is also symmetrical with the twofold rotational C2 symmetry and amino acids like aspartate 25, threonine 26 and glycine 27 from each monomer are located on the floor of the active site and each monomer provides a flap to act as a sealing. The enzyme has a broad substrate specificity, can cleave a variety of peptide bonds in viral polypeptide, but crucially it can cleave with bonds between a proline residue and aromatic residue phenylalanine or tyrosine. The cleavage of the peptide bond next to the proline is unusual and this do not occur in the mammalian proteases such as rennin, pepsin and capsaicin D, and so the chances of achieving the selectivity against the HIV protease over mammalian proteases are good. Moreover, the symmetrical nature of the viral enzyme is active site is not present in the mammalian protease, again suggesting the probability of drug selectivity. The examples of the HIV protease inhibitors are the indinavir, nelfinavir, ritonavir, saquonavir, and emprinavir. So these are the structures are a little complicated. You do not need to memorize the structure, but this is very important. The structure, the active site, and how it differs with the mammalian protease, and why it is possible to achieve the drug selectivity. These things you have to understand and you have to remember. Okay. So I hope it is clear. If there is any doubt, please let me know. And I am going to the next question. But if you have any doubt or query, let me know in the chat box. Okay. So problem number 15 says acyclovir. The structure shown here is an inhibitor of the DNA polymerase selectively in the virus infected cells. This is because number A, acyclovir is first converted to monophosphate only by the viral thymidine kinase. Number B, acyclovir does not enter the infected cells. Number C, acyclovir first converted to the monophosphate only by the host thymidine kinase. Number D, acyclovir triphosphate is not accepted by the host DNA polymerase. Which of the following is the correct answer? Let me know. Your options are number A, acyclovir is first converted to monophosphate only by the viral thymidine kinase. Number B, acyclovir does not enter the infected cells. Number C, acyclovir first converted to monophosphate only by host thymidine kinase. Number D, acyclovir triphosphate is not accepted by the host DNA polymerase. What is the reason? What is the reason that it is a selective DNA polymerase inhibitor only in the virus infected cell? Again, I will wait for a minute or two, then I will check the chat box. Let me know A, B, C, or D, which of the following is the correct answer for problem number 15. Acyclovir is an inhibitor of DNA polymerase selectively in the virus infected cells. This is because, what is the correct reason? Let me check the chat box if there is any answer for problem number 15. Okay, I got one answer saying number C. Now let me check if number C is the correct answer for our problem number 15 or it is not. So the correct answer will be, it is number A. Acyclovir is first converted to monophosphate only by the viral thymidine kinase. Okay. So acyclovir, also known as the which is known as the acyclovir, is widely used antiviral modification medication primarily employed to treat infection caused by various herpes viruses, including the herpes simplex virus or HSV type 1, type 2, responsible for the cold sores and genital herpes, and varicella zoster virus or VZV, which cause the chickenpox or shingles. Okay. Mechanism of action: acyclovir is synthetic purine nucleoside analog that selectively targets viral DNA replication, viral activation. Acyclovir enters the infected cell and selectively phosphorylated to the acyclovir monophosphate by the viral thymidine kinase, an enzyme present only in herpes infected cells. Cellular conversion: host cell kinases then convert acyclovir to monophosphate, acyclovir monophosphate into acyclovir triphosphate and the active form of the drug. Okay, and the DNA polymerase inhibition (ACV), this acts as a competitive inhibitor of the viral DNA polymerase, the enzyme responsible for virus to this also is incorporated into the growing viral DNA chain, terminate and further prevent the viral virus synthesis. Okay, so acyclovir is prescribed for various conditions caused by the herpes virus, including the herpes simplex virus and varicella zoster virus like chickenpox and shingle. And prevention: it can also be used to suppress the therapy to prevent frequent outbreaks of the genital herpes and then these things. Okay. Then common side effects, the acyclovir includes nausea, vomiting, diarrhea, headache, and this are less common side effects that can include kidney problems and neurological symptoms like hallucination, confusion, and allergic reaction. Important consideration: acyclovir manages symptoms and slows viral replication, does not cure the herpes infection, and virus remain latent in the body. Hydration is the crucial during treatment, especially with oral and IV forms, to prevent the kidney-related side effects. Okay. So this is important. Acyclovir enters the infected cell and selectively phosphorylated to acyclovir monophosphate by the viral thymidine kinase. Okay. This is the reason why this particular this is selective for the virus infected cells. Okay. Now let me go to the next problem. Problem number 16 says, which of the following statement is false regarding the admittance? Okay. Your options are: number A, they are known as ion channel disruptors. Number B, they inhibit the replication of influenza A virus by blocking the matrix protein channel. Number C, with a change of concentration, their mechanism of action changes. Number D, virus can't mutate with in the presence of the amantadine. Which of the following statements is false regarding the adamantins? Your options are: number A, they are known as the ion channel disruptors. Number B, they inhibit the replication of the influenza A virus by blocking the matrix protein channel. Number C, with the change of the concentration, their mechanism of action changes. And number D, virus cannot mutate in the presence of the amantadine. Again, I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D, which of the following is the correct answer for problem number 16. Read all the options very carefully and identify the statement which is false regarding the adamantins. Your options are number A, they are known as the ion channel disruptors. Number B, they inhibit replication of the influenza A virus by blocking the matrix protein channel. Number C, with the change in the concentration, their mechanism of action changes. And number D, the virus cannot mutate in the presence of the amantidine. I will wait for a minute. Then I will check the chat box. Okay. Which of the following statements is true regarding the adamantins? Let me check the chat box. Okay, I got an answer which is saying that number C is the correct answer. Let me check if number C is the correct answer or not. So the statement which is correct means the statement which is false regarding the adamantins will be, it is number D. The virus cannot mutate in the presence of amantadine. This statement is false. Okay. So ion channel disruptors or adamantins. So adamantins were discovered by random screening and are the earliest antiviral drugs used in the clinically against flu, decreasing the incidence of the disease by 50 to 70%. Amantadine and rimantadine. So this is the structure of the amantadine and this is the structure of rimantadine. We both are the examples of the adamantins. Okay, are related to the similar mechanisms of action and can inhibit viral infection in two ways. At low concentration, they inhibit the replication of influenza A virus by blocking the viral ion channel protein called matrix M2 protein. At a higher concentration, like about greater than 50 microgram per ml, the basic nature of the compounds becomes important and they buffer the pH of the endosomes to prevent the acidic environment needed for the HA to fuse with the viral membrane with the that of the endosome. These mechanisms inhibit the penetration and uncoating of the virus. Unfortunately, the virus can mutate in the presence of the amantadine to form the resistant variants. Amantadine binds to the specific region of the M2 ion channel. Then resistant variants have the mutations which alter the width of the channel. Research carried out to find the analogs which might still bind to these mutants proved unsuccessful. Work has been carried out in the attempt to find an analog which might affect the ion channel and pH levels are comparable concentration. This has focused on secondary tertiary amines with increased basicity as well as alteration of the structure by reduced activity of the ion channels. The rationale is that the resistant flu variants are less likely to be produced if the drug acts on the two different targets at the same time. Rimantadine was approved in 1993 when less toxic alternative to amantadine for treatment of influenza A. Unfortunately, neither agent is effective against influenza B virus, does not contain the matrix M2 protein. Side effect is also problem, possibly owing to the effects of the host cell ion channels. Okay. So this was all about the adamantins. So the statement number A, which says they are known as the ion channel disruptors, it is true. They inhibit replication of influenza A by blocking matrix protein channel. It is also true. With the change of concentration, their mechanism of action changes, which is also true, which we have seen here that at the low concentration, they inhibit the replication of influenza A virus by blocking viral ion channel protein. And in the higher concentration, the basic nature of compounds become important and the buffer the environment these mechanisms. So these are two different mechanisms at the higher and lower concentration. But the statement which says the virus can't mutate in the presence of amantadine, this is false. The virus can mutate in the presence of the amantadine also. Okay. Let me go to the next problem. Problem number 17. It says which of the following is the correct set of enzymes for the transformation mentioned below. So from this to this, what is the correct set of the enzymes? Your options are number A, thymidine kinase followed by esterase, number B, esterase followed by cell kinase, number C, thymidine kinase followed by aldehyde oxidase, and number D, esterase followed by the aldehyde oxidase. Which of the following is the correct set of enzymes for the transformation mentioned here? This transformation, which is the correct set of enzymes. Number A, thymidine kinase followed by esterase. Number B, esterase followed by cell kinase. Number C, thymidine kinase followed by aldehyde oxidase. Number D, esterase followed by aldehyde oxidase. Which is the correct set of enzymes? Which of the following is the correct set of enzymes for the transformation? Thymidine kinase followed by esterase, esterase followed by cell kinase, thymidine kinase followed by aldehyde oxidase, or esterase followed by aldehyde oxidase. Let me wait for a minute. Meanwhile, let me know A, B, C, or D, which of the following you think is the correct answer for problem number 17. Which of the following is the correct sets of enzyme for the conversion? Let me check the chat box. Let me see if there is any answer for our problem number 17. Okay, I got one answer again, which is saying that number C is the correct answer. Let me check if number C is the correct answer or not. So the correct set of enzymes will be, it is number D, esterase followed by the aldehyde oxidase. Okay, so first is this. You can say the conversion was given like this to this. Okay, so this is the pencyclovir. Okay, so first is the esterase. Esterase converts this esterase actually hydrolyzes these esters to form the alcohol hydroxyl groups. Then we have aldehyde oxidase. Aldehyde oxidase which oxidizes this C double bond N to this NHCO. And okay, so, and then pyryl thymidine kinase can act. So these are the other conversions. Okay, which happens when there is a viral thymidine kinase. What will happen? Monophosphorylation happen, and the cell kinases with the help of the triphosphorylation happens. This is similar to our for what we have seen in our case of our acyclovir, right? So now let me go to the next problem. Problem number 18, it says again, which of the following statements is false regarding the interferon? Again, it is a true false kind of question. So read the options very carefully and then answer. Okay, so which of the following statement is false regarding the interferon? Your options are number A, they are classified as a broad spectrum antiviral agent. Number B, they are small natural proteins. Number C, beta interferons are generated from the lymphocytes. And number D, immunomodulators are used to induce the interferon production. Which of the following statement is false regarding interferons? Your options are number A, they are classified as a broad spectrum antiviral agents. Number B, they are small natural proteins. Number C, beta interferons are generated from lymphocytes. Number D, immunomodulators are used to induce interferon production. I will wait for a minute, then I will check the chat box. A, B, C, or D. Read the options very carefully and let me know which will be correct answer for problem number 18. Which of the following statement is false regarding the interferons? Number A, they are classified as a broad spectrum antiviral agent. Number B, they are small natural proteins. Number C, beta interferons are generated from lymphocytes. And number D, immunomodulators are used to induce interferon production. Let me know. I will wait for a minute. Which of the following statements is false regarding interferons? Let me check the chat box. Okay, got an answer. Let me check if it is. Let me check if it is correct or not. Okay. So the correct answer will be. It is number C. Yes, it is correct answer. Very good. Beta interferons are generated from lymphocyte. This is the correct answer. So interferons are small natural proteins that were discovered in 1957 and which were produced by host cell as the response of the foreign inhibitors. Once produced, interferons inhibit protein synthesis and other aspects of viral replication in the infected cell. In other words, they shut down the cell. It can also be described as an intracellular immune response. Administering interferons to the patients had been seen as a possible approach to treating flu, herpes, hepatitis, herpes, and colds. There are several interferons which are named according to their source, like alpha interferon from lymphocyte, beta interferon for fibroblasts, gamma interferon from the T cell, and alpha interferon, or known as alpharon. Now, IFN alpha is the most widely used of three types. In the past, it was difficult and expensive to isolate interferons from the natural cells, but the recombinant DNA techniques allow the production of the genetically engineered interferons in the larger quantities. Recombinant alpha interferons is produced in the three main forms: alpha 2A, alpha 2B are the natural form. Alpha con 1 is the unnatural form. They have produced successfully therapeutically but can be serious toxic side effect. At present, alpha interferon are used clinically against the hepatitis B infection. It is also used by the with the ribavirin against the hepatitis C infection. Interferon production is the body can be produced by the agents known as the immunomodulator. One such example is the avridine, which is used as the vaccine adjuvant by the treatment of the animal disease such as the foot and mouth. Imiquimod is also induces the production of alpha interferon as well as other cytokines that stimulate the immune system and is the effective against the genital warts. So, antiviral action of interferons, the viral RNA from an infected virus enters the cell. Then the infecting virus replicates and into the new viruses. When the infecting virus also induces the host cell to produce interferon mRNA or IFN mRNA, which is translated into the alpha and beta interferons. Interferons are released by the virus infected host cell to bind the plasma membrane or the nuclear membrane receptors on the uninfected neighboring host cell, inducing them to synthesize antiviral proteins or AVPs. This induces oligoadenylate synthesis and protein kinase. New virus is released by the virus-infected host cell and infect the numerous host cell, but AVPs degrade the viral mRNA and inhibit the protein synthesis, thus interfere with the viral replication. So, as you can see, this is a similar the viral infection, but this viral infection this induces the interferon mRNA, which gets released and it actually induce the neighboring host cell, which is a healthy cell, to synthesize antiviral protein. So now when this cell will be infected, these antiviral proteins will degrade the mRNA, viral mRNA, and now the then further viral infection it is stopped. So this is the antiviral effects of the anti-interferon. Okay. So now let me go to the next problem. Problem number 19, it says that infovertide is the first fusion inhibitor targeting HIV, matches with the viral protein dash. Fill in the blanks. Your options are number A GP45, number B GP40, number C GP41, and number D GP31. Infovertide is the first fusion inhibitor targeting HIV, matches with the viral protein dash. Fill in the blanks. Whatever A, B, C or D, which of the following is the correct answer, let me know. I will wait for a minute because we don't have much time and I have to discuss one more question today. Enforvatide is the first fusion inhibitor in targeting HIV matches with the viral protein, which is the correct answer. GP45, GP40, GP41 or GP31? Let me check the chat box. Is there any answer for problem number 19? Okay, I got two answers. One is saying number B, other is saying number A. Let me check which of the following will be the correct answer for problem number 19. So the correct answer will be, it is number C, GP41. Okay. So other agents that are under study for the treatment, okay, for the other agents that are under study for the treatment of HIV include the integrase inhibitors and cell entry inhibitors. Blocking entry of the virus into the host cell is particularly desirable because it is so early in their life cycle. Enforvatide was approved in March 2003, was the first member of new class called fusion inhibitors. These are the polypeptide consisting of 36 amino acid which matches with the C-terminal end of the viral protein GP41. It works by forming an alpha helix and binding to a group of three similar alpha helices belonging to the GP41 protein. The association prevents the process by which the virus enters the host cell. In order to bring about the fusion, the GP41 protein anchors the virus to the cell membrane of the host cell. It then undergoes the conformational change where it binds a grouping of the six helices using the three already present in the focus for the grouping. This pulls the membranes of the virion and host cell together, permit fusion. By binding to the group of three helices, enforvatide block the formation of the required hexamer and prevent the fusion. The manufacture of enforvatide involves 106 steps, which is very expensive and may limit the use. A smaller compound BMS 3788006 is being investigated for the binding of the GP120, prevent the initial binding to the virus to the CD4 cell surface. So this is the virus and this is the host cell, and GP41 is the reason why this both can fuse. And so but this particular enforvatide, it binds to the GP41 and it actually acts as a fusion inhibitor, and which inhibits the virus and host cell fusion, and that's how it exerts its antiviral activity. Okay, now last question of today's session, it says that oseltamivir is a dash agent. Your options are number A, anti-influenza, number B, anti-herpes virus, number C, antiretroviral, and number D, antifungal. Oseltamivir is a dash agent: anti-influenza, anti-herpes virus, antiretroviral, or antifungal. Let me know A, B, C, or D, which of the following is the correct answer for our problem number 20. Oseltamivir is which type of agent? Anti-influenza, anti-herpes virus, antiretroviral, or antifungal agent? Oseltamivir is a dash agent. What will be the correct answer? Since we have to discuss it, let me check the chat box if there is any answer for the problem number 20. Okay, I got answers. One is saying number D and another is saying number C. Let me check number D or number C. Which of the following is the correct answer, or is there any other correct answer? So the correct answer will be, it is number A, anti-influenza. So oseltamivir is an anti-influenza agent. Okay. Okay, let me just highlight the things. So this is the life cycle of an influenza virus into the host cell. Okay, so this is the similar to the life cycles of the other. I am not going to just repeat it. So the steps are like absorption, and then the endocytosis happens here. Okay, and when the first the endosomes are converted to the, after uncoating it forms a single strand RNA, and then translation and via this via single strand RNA enters the inside the cell, and then translation it gives rise to the viral proteins and capsid and nucleosides, and then budding and then release virus protein incorporated to the cell membrane. So the life cycle of the influenza virus inside the host epithelial cell are almost similar to what we have discussed. So oseltamivir is used to prevent prevention to treatment of the influenza causes by influenza A or B viruses. It is a complementary list of World Health Organization list of essential medicine as the most important medication needs for the basic health system. As per December 15, 2010, WHO has reported 314 samples to prevent 2009 H1N1 flu tested worldwide show to resistant to oseltamivir. The vast majority of mutation confer to the resistance of single amino acid mutation in the neuraminidase enzyme. The meta-analysis of 15 studies have pulled the incidence rate of oseltamivir resistance by 2.4%. Subgroup analysis detected higher rates among the influenza A patients, specifically H1N1 prototype. So this is the structure of oseltamivir, which is also known as the Tamiflu, and which is a very crucial anti-influenza drug. So this was the last question of today's session. So today basically we have discussed regarding our various antiviral agents. Okay, and so mostly we have discussed about antiviral agent. We have discussed about the virus life cycle, and also we have discussed about the polyketide biosynthesis and the enzymes responsible for it. So this is my email ID. So if you have any doubt or any query, please let me know via email. Okay, you can also put your queries in the discussion forums of the NPTEL SWAYAM website, where if you have any query regarding the any problem solving sessions, you can post your queries there also. I will try to solve your queries via via the discussion forum. So if you have any doubt regarding today's class, today it was our week 11 live session. We will meet on the next Friday, which will be our last live session, our week 12 live session, because it is a 12-week course and last week's content has already been released. So next Friday we will have our last live session, and after that I think we will have a summary session of 1 hour that will be notified to you via email. Okay. So I will close the session for today if there is no doubt. If there is any doubt, you can let me know in the next class or via email or via discussion forum. So thank you all for joining. Thank you all for answering in the class. I will meet you on the next Friday, and I am closing the session for today. Okay. Thank you.